past 5 years than during all of human history combined. This knowledge has been driven by advances in technology. The decoding of the human genome allows us to look further inside our brain cells than ever before, providing a window on how genes dictate brain function and behavior. Developments in scanning technology also offer scientists unprecedented access into brain activity. New discoveries could someday promote mindfulness, increase cognition, and offer treatments for disease. What do people like you, who have spent your life dedicated to understanding brain and mind, understanding first the psychiatry and then later the science of the brain?
Mind is a series of functions carried out by the brain. Every single thing that you and I do, every aspect of our behavior is carried out by the brain. From the most simple reflex act, hitting a tennis ball, running, riding a bicycle, to the most creative aspects, running a Charlie Rose program. These things are all carried out by the brain. But in the first 19 centuries, people were limited to the tools of philosophy. Then psychology began to be a powerful discipline. Then brain sciences came along. And what's happened in the last few decades is these fields have come together.
You were trying to bridge them together.
Many of us. You and I are going to do this in this program. But what the field has done, it has brought psychology, brought brain sciences, brought molecular biology, brought this all together. And just like we focused on the gene as being the target of biology in the second half of the 20th century, there's every reason to believe that the mind is going to be the main focus of biological sciences in the 21st century. Consciousness is an interesting question because I cannot determine what is going on in your head. Why you feel a particular way about something is something only you can experience, only you know about. And there is no way that I have immediate access to that. Now, maybe someday we'll have a way of visualizing neural correlates of consciousness, but at the moment, we don't have that.
Now, here's one of the things about the brain that I've always found fascinating. It's always changing.
That's right.
My little brain will be different at the end of this conversation than it was at the beginning.
And after the whole series, you'll be a completely different person.
But everything we do makes the brain different.
Absolutely.
You know,
Absolutely.
Drinking this water
Absolutely.
makes it a different brain. It gives me a different experience.
This not only explains how we grow intellectually, but it also explains the enormous capability the brain has to recover from damage. This is also another reflection of plasticity of the brain. We also will consider that in this series.
So in the end, I may come out of this with my brain changing so much I want to wear a bow tie.
That'd be wonderful. I'd be glad to give you
have
one.
a collection, do you?
What we have here is the beginning of a very brief tour of the structure of the human brain. We're looking at a computer-generated image of the left hemisphere of a human brain. Now, this brain is covered, as the human brain is, most prominently by a structure called the cerebral cortex, which is this gray mantle of tissue that's really almost all that's visible when you look at the human brain from the side. And the cerebral cortex is the main computing machinery of the brain, and it's also the part of the brain that's most highly evolved in humans compared to our ancestors in the animal kingdom. Now, we customarily divide the cerebral cortex into 4 lobes, and the 4 colors in the image here show you the 4 lobes and their positions. And the 4 lobes have many different functions, but we characteristically think of them as having a number of sort of primary functions. The occipital lobe here, shown in red at the back of the brain, is the main seat of visual function. That's a lobe which is particularly dear to my heart because it's the system of the brain that I work on. The temporal lobe, here shown in green, is involved in a number of complex functions. It's involved in higher processing of visual information. It's involved in the laying down of memories. And it's involved in the processing of information about sounds and in the conversion of information about sounds into linguistic representations and into the programs that allow us to speak and understand language. The parietal lobe, here shown in blue, is sort of the central spatial orientation organ of the brain. The parietal lobe combines information from multiple senses, including vision, hearing, and touch, and it formats motor commands, output commands, for the muscles, which allow the body to be oriented in space with respect to the stimuli in the environment. So when you navigate in the world, when you look around the world, when you decide what parts of the world to pay attention to, it's the parietal cortex which is doing the bulk of the work. Finally, in purple, here is the frontal lobe at the front of the brain. The frontal lobe is the most highly evolved part of the cortex in humans. It is the part of the brain that is probably most directly responsible for making us human. It is the part of the brain that is responsible for decisions, for actions, for many functions that are often now called executive control functions, deciding what to do, where to go, what to eat, what to say, probably more importantly, what not to do and what not to say and where not to go. And so it's the ultimate sort of seat of central control in the brain. So we're now looking again at the view of the left hemisphere of the human cerebral cortex. Now, the cerebral cortex looks like a cauliflower in this kind of view, and that's the way we tend to think about it. But it is, in fact, a very elaborately organized sheet of cells, which has been folded up so that it actually fits inside the cranium. And a way to visualize that is the following. Imagine that the brain's computing machinery is actually flat, like this piece of paper, but the skull is not well suited to accommodating a flat piece of paper. So what do we do to accommodate it in the skull? Well, we basically crumple it and fold it, and we create a series of folds. And in the next video clip, which was prepared by my colleague David Van Essen at Washington University, what we're going to see is that process in reverse. We're now looking at the right hemisphere, and this is actually a computer-generated image generated from magnetic resonance imaging data. And it's been colored so that the light-colored parts of the image represent parts of the brain near the surface of the skull, and the dark-colored parts of the image represent parts of the brain that are buried deep within these folds, which we call sulci. Now, in the first section of the film, I'll just spin this around so that you can see the three-dimensional structure of what we're looking at, which is, of course, similar to what we've seen before. And in the next segment of the film, what we're going to do is treat this brain as though it were made like a child's balloon out of rubber, and we could inflate it just by pumping air into it. So the brain gradually inflates. The things that were colored dark to indicate their depth in the brain are still dark, so you can keep track of what was deep in a sulcus and what wasn't. But now you have a much simplified form of the brain, what we call a semi-inflated form. Finally, the last stage of this process is to flatten this semi-inflated form out into a completely flat sheet. And now what you can see is that this cauliflower structure that we're used to seeing in the brain as this elaborately folded cerebral cortex is in fact a single flat sheet of cells. Now, this particular sheet shows by its coloring how deep in the brain the structures were. But when we talk in the course of the series, as we will, about localization of function in the brain and talking about different parts of the brain performing different operations, what we'll be talking about is different regions which are most conveniently and most accurately visualized as living on this sheet, the unfolded sheet of the cerebral cortex.
So in the field of genetics right now, there's tremendous excitement because in the past 10 years or so, we have decoded the human genome. And that represents the complete set of instructions for building this marvelous organ and for building into it the capability of generating unique human creative actions. So the reason that all humans can generate language is that humans have special genes that distinguish us from the animals that allow the brain to do things that animals' brains cannot do. And we have now this instruction manual. There are 25,000 genes. It's sort of like having an encyclopedia with 25,000 entries. And we are in a position to try and understand what those entries make possible.
Would you just briefly for a moment consider to what degree behavior is determined by genes versus learning?
That's the essential question. And the answer is that it is that parts of behavior are determined by genetic programs and parts of behavior are determined by experience. And you can point to examples that are almost entirely one or the other. So, for example, our ability to see colors and to distinguish colors is something that is encoded by 3 specific genes expressed within our eyes. that compare blue and red and green to each other and split them up. And if you're missing one of those genes, you're red-green colorblind, and there's no amount of thinking you can do to solve that problem. That gene has determined something about the way you can perceive the world. On the opposite extreme, the fact that some of us speak English and some of us speak Chinese has nothing whatsoever to do with our genes, and it's completely determined by our education and our experience. And furthermore, how well we speak English and Chinese is determined by when we have those experiences.
But I want to know, how does the brain produce this remarkable experience of qualitative, unified subjectivity? See, all the experiences we have, everything, the smell of the rose, the depression you feel at your income tax, the sound of Beethoven, or pick your favorite, the angst of post-industrial man under late capitalism, whatever's your favorite experience, it's all produced by variable rates of neuron firings in the brain. And that to me is the most, that's why I'm here. I want you guys to answer that question for me. The hard problem, and it's hard to phrase this problem exactly, but it's roughly speaking, how does the brain get over the hump from neuron firings to human feelings, to human and animal feelings? Now, people sometimes say consciousness is hard to define, but I think we're just talking about not the analytic definition that comes at the end of the investigation, but the common sense definition that identifies the target. It's not so hard to define. Consciousness is these states of qualitative feeling or sentience or awareness. They begin in the morning when you wake up from a dreamless sleep. They go on all day until you go to sleep again or otherwise become unconscious, and they have these remarkable features. One, there's always a special qualitative feel. Listening to music is different from brushing your teeth. It's just a different qualitative feel to it. Secondly, it's subjective. It's all going on in me, and I'm pretty sure that something goes on in you. It's like what goes on in me, but I have a relation to my own consciousness, which is not like my relation to anybody else's. And third, there's this remarkable feature. It's unified. So I don't just now feel the shirt against my neck and hear the sound of my voice and look at all the people around the table and see these wonderful statues of the brain, but I have them all as part of a single unified conscious field.
What we've learned about memory is that memory involves an alteration in how nerve cells communicate with one another. And that'll become even clearer once Jerry Fishback gives us an outline of how the nerve cells function. Nerve cells communicate, as he will show, through a point of contact called the synapse. And what happens with learning is the strength of these synaptic connections change. In short-term memory, when you learn something very briefly and you forget it in a day or so, it's a functional change. There is no change in anatomy. But if you learn something for the long term by repetition or by listening to this program, drawing all of your attention, there is a change in the expression of genes, not a mutation, but an amplification of how well a gene is functioning, and that leads to the growth of new synaptic connections. So if you remember this program the next day, it is because an anatomical change has occurred in the brain, and we hope that the people listening to these set of programs will walk out of this series with a completely different set.
I've actually done this a bit at a time on a larger board, but I just want to walk you through this. Basically, nerve cells are the building blocks of the brain, and whatever we do to understand the genetic composition or the environmental influence must take these cells into account. Now, nerve cells are cells just like any other cell. There's a cell body, there's a nucleus that contains the genes. The genes encode proteins, hormones, enzymes, structural proteins that are extruded out into the cytoplasm. There are many of these nerve cells in the brain. Some people estimate 100 billion nerve cells, more than there are stars in the Milky Way galaxy. The other thing that's extraordinary about these cells is they generate signals, which will end up at the synapse. And they generate electrical pulses, which are brief, about 1,000th of a second, and about 1/10 of a volt in amplitude. And many people feel that the information in the nervous system somehow is encoded in the frequency or the detailed time structure of these impulses. They're conducted at various rates. And big neurons are conducted at about 100 miles an hour. It's not as fast as an electron. And if you place an electrode near them and amplify the signal, you can actually hear them So our brains are continually active, even when asleep. And all this takes an enormous amount of energy. If blood flow is cut off to the brain for a matter of seconds, these cells will die. Whereas if you cut off blood flow to a tensely contracting muscle, it will survive. It will survive for minutes, many minutes, half an hour without blood flow. But these cells are using much more energy every second of the day and night than our muscle cells when they're contracting. When these impulses reach the end of the axon, they contact the next cell in line at the dendrites by these structures called synapses. And that's what many of us have studied through much of our career. For every nerve cell, there may be up to 1,000 synapses. So 100 billion times 1,000 is a lot of synapses. But I don't believe, I know no one here believes, that's where the complexity lies. The complexity of the nervous system, if we're going to deal with this, lies in how these synapses change as a function of the genetic instructions and as a function of experience, listening, life experiences, and they do change. And as many have pointed out, these synapses grow, they form, they retract. The brain is not hardwired. It is within limits. There is an architecture to the brain, but on the micron scale, there's a great deal of flexibility.
One of the things that I think is very important to keep track of when you have something like this in front of you, we often think of the analogy of the brain as a computer. And in fact, the brain, of course, is an organ whose job is to process information. So it is a computing device. But the brain works the way it does because it's made of meat and not of silicon and copper. And so the fact that these axons transmit impulses at 100 miles an hour is not the same as a computer sending a signal at the speed of light. No. And the fact that each of these axons makes many variable contacts through chemical means with other cells is very different from connections to a transistor or a resistor or an electrical component.
And for a long time, the computer metaphor was really an impediment in cognitive science because it led people to the illusion that the anatomy doesn't really matter. All that matters is the programs. You get the right program. And I think for a whole lot of reasons, that's a very unintelligent view, and I think we're getting out of that view. We have to, and what I like about this particular group is we all respect the anatomical, biological specificity of the brain. It's not an accidental organ. You can't do it with just anything.
So what I'd like to hear Corey just say one more word about is these cells are organized into circuits. And the circuits are very meaningful, and those circuits are organized into larger ensembles. So to go from a gene alteration to a circuit that can explain a simple behavior, I think will be one of the great triumphs of neuroscience.
And that is one of the miracles of the brain, and it was really Eric's discovery and contribution to neuroscience, that an experience in your life, that an emotion that you feel, that a thought that you have is turned into a biological, physical.
We all respect the anatomical, biological specificity of the brain. It's not an accidental organ. You can't do it with just anything.
So what I'd like to hear Corey just say one more word about is these cells are organized into circuits. And the circuits are very meaningful, and those circuits are organized into larger ensembles. So to go from a gene alteration to a circuit that can explain a simple behavior, I think will be one of the great triumphs of neuroscience.
And that is one of the miracles of the brain, and it was really Eric's discovery and contribution to neuroscience, that an experience in your life, that an emotion that you feel, that a thought that you have is turned into a biological, physical reality by your brain. I think the big question is, how does one brain generate infinitely many behaviors? Because we sort of talk about these fixed cells and we talk about these fixed connections, but there's traffic flow through this brain, and the traffic is constantly changing. The traffic when you're asleep is completely different from the traffic when you're awake, and the traffic when you're nervous is different from the traffic when you're relaxed. And so we have one brain and one set of biological structures that can generate infinitely many responses through internally generated activity, through external generated activity. I think that's a great problem.
So I guess implicit
So to go from a gene alteration to a circuit that can explain a simple behavior, I think will be one of the great triumphs of neuroscience.
And that is one of the miracles of the brain, and it was really Eric's discovery and contribution to neuroscience, that an experience in your life, that an emotion that you feel, that a thought that you have is turned into a biological, physical reality by your brain. I think the big question is, how does one brain generate infinitely many behaviors? Because we sort of talk about these fixed cells and we talk about these fixed connections, but there's traffic flow through this brain, and the traffic is constantly changing. The traffic when you're asleep is completely different from the traffic when you're awake, and the traffic when you're nervous is different from the traffic when you're relaxed. And so we have one brain and one set of biological structures that can generate infinitely many responses through internally generated activity, through external generated activity. I think that's a great problem.
So I guess implicit in much of what we've talked about today is the challenge that I think is the one that really faces us in all of these things. It faces us in answering John's question and in Jerry's question and in Corey's question. And that's the challenge of identifying what level of brain function we should consider when we try to answer any of these questions. The brain's unique among organs in that we have to consider its molecular structure, its cellular structure, the way the cells communicate, the way they make circuits, the way the circuits make areas, the way the areas make lobes. And each of those levels of analysis has a level of answer associated with it. The trouble with answering John's question or Jerry's question is I don't think we know even what level to ask the questions at. And so although we have tools that let us approach the questions at each of those levels, we could be sniffing around at an altogether incorrect level in trying to answer a question that's really answered at a molecular level or at a circuit level or at an area level.
The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation,